Welcome friends, welcome to Datta Biology class. So let us continue our discussion on transcription. Today will be the transcription 4. I think this will be the concluding part of the transcription and then we will be thorough with transcription. So before starting transcription 4, we have completed the other things, that is amino acids. We have completed how the amino acids are there, and then we have completed mRNA, RNA, all RNA, amino acid, RNA. Now let us know something about ribosome. Ribosome, the protein factor. Okay. So a little bit about ribosome will give you much more better idea of the transcription process proper. Now ribosome consists of two parts. We are starting with ribosome a little bit. Ribosome consists of two parts. Two unequal parts consist a ribosome. This is the smaller one. This is a little bigger one. And connecting these two, it will be a further bigger one. Here, now we know that ribosome consists of two unequal parts, the smaller one and the bigger one. In prokaryote, this is made up of 30 eggs. In eukaryote, it is made up of 40 eggs. In prokaryote, it is 50 eggs. In eukaryote, it is 60 eggs. In prokaryote, jointly it is 70 eggs. In eukaryote, it is 80 eggs. Here, 30, 40, 50, 60, 70, 80. No need of taxing your brain. Simply write prokaryote and eukaryote, smaller, bigger and much bigger. And write 30 years, 40 years, 50 years, 60 years, 70 years, 80 years. Now the question comes, why 30 and 50 years is equal to 70 years? And why 40 years and 60 years equal to 80 years? Now this S is known as Wedward unit. S identifies or denotes Wedward unit. Okay. It is sedimentation coefficient. This is actually sedimentation coefficient. Now, if you take 30S in a centrifuge, in a test tube, and put it in a centrifuge machine, the sedimentation, it gets sedimentation and it is marked as 30S. Say for example, within 30 minutes it gets sedimented. And if you take this along, the sedimentation coefficient will be 50. And jointly, if you take jointly these two, it will have sedimentation coefficient of 70s. That's why it is known as 30 plus 50 is equal to 70, 60 plus 40 is equal to 80. To keep it in mind, write in this way 30, 40, 50, 60, 70, 80. Clear? This is ribosome. Ribosome has two units, that is smaller and bigger, 2 units. In prokaryotes, I am concerned with the prokaryotes now, not with this. In prokaryotes, this 30S this is 30S ribosome. This 30S ribosome contains one RNA. This RNA is called 16S RNA. Clear? In prokaryotes, these 30S smaller unit of ribosome contains one 16S RNA. And now what actually happens? In prokaryotes, the mRNA is like this. mRNA has codons complementary to the DNA from where it is produced. So these are the codons. And it starts from 5 prime to 3 prime. If you start from 5 prime to 3 prime, you will get AUG, the initiation codon, the initiator. Now AUG is here, and astonishingly, you will get a bunch of nucleotides here. A bunch of nucleotides here. These nucleotides are rich in purine bases. What is purine bases? Adenine and guanine are purine. Thymine and cytosine are pyrimidine bases. This is purine, pyrimidine. And in RNA, this thymine is so in prokaryotes, in 30S ribosome, we get a special 16S RNA along with some other protein factors, say 20 or 21 proteins take parts, 20 or 21 proteins factors are there. Now what I said, if you start reading from 5 prime to 3 prime of mRNA, this is mRNA. mRNA. You will get the initiation codon. Now if you go upstream again, you will find a special sequence of nucleotides which are purine bases. Purine bases means A, G, G, A, G, G, A, G, G. This way, the triplet codes are there. But in the mRNA or prokaryotes, they do not get translated into protein. They do not get translated into proteins, and this area is known as Shine-Dalgarno sequence. They do not get translated into protein. Actually, this is a sticky area. They act like anchor. Anchor means, in anchor there are some hooks. In anchor, we have seen in seaport. These anchors, they act like anchors or hooks. They are rich in purine bases like this. This sequence is known as Shine-Dalgarno sequence, named after the discoverer of this sequence, Mr. Shine and Mr. Dalgarno. And sequence is not the name. So this is Shine-Dalgarno sequence, which is a sticky area, and it anchors like it acts as an anchors or act like hooks during protein synthesis, and this area is full of purine. They do not get translated. Why? It is seen that in the 16S RNA, they are also 3' to 5' remains a sticky area. This is full of Purine pyrimidine bases. So this is purine bases, this is pyrimidine bases. This is known as, this sequence is known as anti-Dalgarno sequence. Clear? This was not known to us. Actually, what happens? When this mRNA comes out, it remains here like this. It cannot get attached anywhere. So this is a point of attachment with this to this, so as to align the initiator product in the P site, which I will come later. So to give this initiation, to keep the initiation codon in proper place, this they get stitched here. This along with 20 to 21 proteins, this 16S ribosome contains an anti-Dalgarno sequence where this gets sticked. Clear? The two subunits, here the bigger subunits I have not written, the bigger subunits is there. They remain scattered in the cytoplasm. Why I am saying that this Shine-Dalgarno sequence is not available in eukaryotes. Why? Let us, this is eukaryotic cell, say this is prokaryotic cell, definite nucleus, and there are double walled endoplasmic reticulum. While here in prokaryotic cell, there is no endoplasmic reticulum. So these ribosomes floats here and there, and now this mRNA will not find any particular place where they will join. They come out, they already been prepared within the cytosol. Now to get themselves attached to the particular place to form the protein, so this arrangement has been made in prokaryotes. So in prokaryotes we got the Shine-Dalgarno sequence and anti-Shine-Dalgarno sequence where the mRNA attaches itself with the smaller unit, with the 16S ribosome or 30S ribosome of the smaller unit of ribosome. Clear? Here, since there is endoplasmic reticulum, the ribosome remain attached here in endoplasmic reticulum. So you have come across that whenever there is ribosome, it is called rough endoplasmic reticulum, and if it is smooth, it is called smooth endoplasmic reticulum. So within the endoplasmic wall of the endoplasmic reticulum, the ribosomes are situated in eukaryotic cell. Clear? This is all about RNA. So after completion of this ribosome, we go directly into the transcription process. Then it will be easier for you to understand. Already we have discussed everything. Now a little bit time to understand it properly. We should difference number one. We will write while transcription process will come, formation of mRNA. That we will have to go through. We will have to write. And then number two, we will go in the cell. There are amino acids that has to be activated. So there is the activation of amino acid. We will say activation of amino acid. Part 2. Number 3. Now this activated amino acid will get touched with the tRNA. So attachment of amino acid with tRNA. Number 3. Then it will go to ribosome where protein will be produced. So we will write initiation of polypeptide chain. There polypeptide will come. We are coming. Initiation of polypeptide chain will be 4. Number 1, assembly of polypeptide. Polypeptide. Assembly of polypeptides. This much will be enough. So the core process of transcription will make it shorter and will make it comprehensive, writing all these 5 stages, simple steps, we will write it. Number 1, we will write that during the process, formation of mRNA, we have come across But what we will write? We will write in a short way as per the number given in the question paper. In the question paper, if it is given 10, we will write as per 10, or if it is 5, we will write accordingly. So, formation of mRNA we will have to write. In case of formation of mRNA, we will write there is from DNA mRNA are produced. So there is a unwidening of this. Clear? This is, it gets wide. So it gets wide. Not in the case of DNA replication, we have said there are helicase enzyme, there is not. There is a localized unwidening of the DNA DNA strand. So number one we will write unwidening here. Unwidening of the strand, DNA strand, localized, and it is not reflected to the entire length. Localized unwidening, and this localized unwidening, there remains the two strand, number one strand and number two strand, and the transcription. And the transcription will start from 5 prime to 3 prime, 5 prime to 3 prime, which we have come across. So this strand will be 3 prime to 5 prime. Clear? So this is known as same strand. So we will write something about the after unwinding. This starts from 5 to 3 end. So we will write 5 prime to 3 prime. mRNA will be produced from 5 prime to 3 prime. Otherwise, in other sense, you can write from the 3 prime to 3 prime, the mRNA starts, the 3 prime of that is DNA strand. Or you can say that mRNA starts from 5 prime to 3 prime. Now this position we have come across. Clear? This will be here, it will be complementary to this. Only the difference is thymine will be replaced by uracil and the deoxyribose sugar will be ribosome. This much we will write. So this strand is known as same strand. Same strand, and the opposite one, this strand will be called as nonsense. Nonsense strand. This is acting as a template from which the mRNA will be produced. So this strand is known as template strand. Clear? Don't write anything which may distort our attention and mind. Simply say there is a localized unwidening of the DNA strand. One of the DNA strand acts as a template called the template strand or same strand, and the mRNA production proceeds from 5 prime to 3 prime. Likewise, contrary to that, the DNA should have 3 prime to 5 prime. This is called sense and nonsense strand. This much will be enough for writing because you are getting 5 to 6 marks. Now, this portion to this portion is known as coding region. You please mention A, B, C, D, coding one by one. You start thinking and write in that. That this is known as, this portion is known as coding region. Here, now, as soon as this mRNA is formed, what actually happens? This mRNA is formed already here. So we will write, as soon as the mRNA is formed, it is not here. It has localized unwinding, so it will recoil again. After this stretch, after this short piece is formed from the DNA, so from the DNA it opens, produces and recoils again. So we should write that as soon as the mRNA is formed, there is recoiling. This is inflating the subject, nothing more. So there is a localized widening followed by recoiling. So widening has gone. So it will be in its original shape. It will be in its original shape again. Now this one molecule can produce, DNA can produce so many mRNA that I have done. So mRNA is produced. So there is recoiling followed by the which will behind that it will go and preparing this 5-3 so many are produced. So followed by, we will say behind the newly, and then we will say behind the newly formed, recoiling behind the newly formed mRNA. So it will recoil again. Not that it will recoil from here, it will recoil here. It is behind this mRNA formation, behind the coding region also. Behind the coding region it gets. Now as soon as this is produced, mRNA is produced. Now when mRNA is produced, it do not directly goes into the cytoplasm. It do not directly goes into the cytoplasm because it undergoes processing, slicing and others that you will learn in the higher classes. Slicing is done because the bread we make it slice, so slicing and some other factors are there. So as soon as it is produced, it undergo processing. It undergo processing, further processing, and come out in the ribosome, touches the ribosome. Now this ribosome has initiated codon, first codon, second codon, third codon, in this way. Likewise, we will write FG. It has got initiation codon, say for example initiation codon here, AUG. So initiation codon here or here, initiation codon is here. It has got initiation codon. These as soon as this produced, it undergo processing and in case of eukaryotes it say goes out to cytoplasm, it attaches itself to smaller subunit of RNA. So here we know that These are complementary to this AUG and it will attach in the second case. It will attaching itself. And now you can write also one thing. Whenever it is producing the some polymerase, RNA polymerase is required, right? RNA polymerase enzyme helps for the synthesis of mRNA. Along with, you can write initiation factor 1, initiation factor 2, initiation factor 3. They are also protein, they help in this. So these are the point 1, 2, 3, 4, 5, 6, 7 points you have already done, given. Now we come to the next, activation of amino acid. Clear? Formation of mRNA, we have come across, and finally we will write these points. We will take it down, write in this way, in a seriatim. Now, activation of amino acid. Number 2. Activation of amino acids. There are how many amino acids? 20 amino acids. So, there are 20 amino acids should be there. Leucine, isoleucine, phenylalanine, tryptophan, valine, threonine. In this way, there are 20 amino acids in the cytoplasm. So they remain dormant. They remain sleepy. They has to be activated to attach themselves with the tRNA. So this has to be activated. There comes the enzyme. There comes an enzyme. Clear? So there should be 20, minimum 20 enzymes. These enzymes are called, here you write, amino acid tRNA synthetase. So with the help of enzyme amino acid tRNA synthetase, they gets activated. You can write simply amino acid synthetase or amino acid. It should be named as amino acid activatase. But they are given name. We cannot go further. So amino acid tRNA synthetase helps to bind this amino acid with the amino acid tRNA synthetase, SYNTHETase. So they get spun. Amino acid tRNA synthetase. So here what happens? Amino acids binding with this. This is known as amino acid tRNA synthetase. Amino acid tRNA synthesis. They activates it. And now the reaction, basic reaction, you should write here. Amino acid with amino acid tRNA, ATP is required here. Write in this way. amino acid tRNA synthetases. What is produced? It gets active. So amino acid, it is getting active, and this ATP, this joins with AMP. This is formed, and along with this pyrophosphate is formed. Very difficult. Let us clear it. Now, amino acid, ATP, adenosine, sugar, phosphate, phosphate, phosphate. So adenosine triphosphate, and here AMP, adenosine monophosphate is there. So this is adenosine monophosphate is this, and these two phosphate called pyrophosphate, they supply energy, and it along with the amino acid monophosphate, this is known as amino acyl adenylate, or simply amino acid AMP. So that we will write amino acid AMP. So ABC, you write amino acid AMP, adenosine monophosphate, or you can write amino acid adenylate. So this has become amino acid adenylate. Clear? And this is known as pyrophosphate. So it gets activated. It was sleeping. Now suppose it is a school girl. She was sleeping and she has to go to school. So you have to prepare him. So you have prepared the bag, school bag, and the school bag you have given tiffin, this, pen, pencil and other, and you have fixed this school bag into her bag. So this is active. Now it is activated, it can go anywhere. Clear? So this much you will write in case of activation of amino acid. Clear? Only 1 point, 2 point and 3 point. And it's produced amino acid adenylate or amino acid adenosine monophosphate, or simply write amino acid AMP, because sometimes in some books it is written amino acid AMP, sometimes thereafter they are writing adenylate, this and that, nothing. Simply keep it in mind AMP. This AMP, don't write adenylate. Since AMP, it got activated. Clear? Now attachment of this activated amino acid, you should write AAA. Then it will be clear. Attachment of activated amino acid with tRNA. Now this activated amino acid will join with tRNA, which will collect. They are acting as packers and movers. So number 2 I am erasing. Very simple. It's not difficult, but in system you have to write. Then it will attach with activated amino acid attachment with the tRNA. Here in the cytoplasm there are tRNA. So suppose this is tRNA. Suppose this is tRNA. This tRNA can hold only one amino acid. So this is a school bus. School bus. This is for one amino acid to sit. In the school bus there are another amino acid to seed. These are the seed. These are seed. Seed. In this seed, they will sit. This is the back. This seed. This is a seed. So school bus there are 6. But school bus moves as a whole and it moves singly. So we can say it cannot be termed as school bus. You write this in a way. This is garbage. This is tRNA. So in this tRNA, it will sit. And it will carry this to the ribosome. So here what happens? The same enzyme, amino acid tRNA synthase, remain tightly attached with the bird or with the amino acid, and it comes to tRNA, specific tRNA. Suppose this amino acid is alanine, so this tRNA should have the capacity to take alanine, because alanine is much bigger in size, so it will be bigger in size, and proline is smaller in size, so it will be smaller in size. So as per the capacity, they will take. Now this amino acid will come here, attachment. This amino acid will see amino acid. Now this will be called amino acid AMP हो गया. This is tRNA. This is tRNA. This tRNA, amino acid comes here and then it goes out. Amino acid fits here with the respective tRNA, and this is the same enzyme which is binding this schoolgirl and throwing this schoolgirl into this vat or into the bus to go for school. So this amino acid tRNA synthetase is gone now, and now it has become very sticky question. It is known as amino acid tRNA complex. Very important point. We should not forget. So up to this amino acid tRNA synthetase will hold the amino acid T, which is the tRNA, and in the tRNA amino acid tRNA synthesis is done, and this tRNA and amino acid is known as amino acid tRNA complex. This is amino acid tRNA complex. Clear? This amino acid tRNA complex. Now we'll move to specific position of mRNA. Here it is mRNA. In the mRNA there is codons, different codons for different amino acids. So there should be minimum 20 amino acids. There should be 20 amino acid tRNA synthase. Different. There should be 20 types of amino acid tRNA complex. So it is carrying leucine, another one lysine, valine, tryptophan, proline, glutamic acid, aspartic acid. So 20 types of this. So 20 different types of tRNA complex must have to be produced, 20 different types of amino acid to be activated, because we know there are 20 amino acids. Now this amino acid will recognize here where they will sit. So it will sit here, say this is AUG, August. This is known as August. In August, what will be there? In August, reverse will be UAC. This is anticodon, and this tRNA will have which? Methionine. Methionine will be produced. So this is for methionine. This is another tRNA for valine, another tRNA for leucine, another tRNA for isoleucine, as per the direction of the DNA. So it is a small stretch, but I have said that during hemoglobin 574 amino acids are there. So there should be 574 amino acids in this amino. 146 each in mRNA. So this is a big sequence of polymer. So big polymer will be produced. Now here we see attachment of tRNA, initiation of polypeptide chain. Clear? Now we will write initiation of polypeptide chain. Up to this clear? tRNA will bring their respective places. This is for methionine, threonine, this. In mRNA it will join, and it is known as polyribosome or polysome. So they remain attached here in the mRNA. And mRNA fits itself to ribosome, which we have already gone through, but we decide now initiation of polypeptide chain, how it is prepared. 4. Number 4, we will write a different story. Very simple, very simple. There are, this is smaller subunit, of ribosome. mRNA sits, smaller ribosome sits on mRNA. Say this is mRNA. Now what will happen? This mRNA has been produced with the help of some initiation factors and others. So now initiation of polypeptide chain also required initiation factor. So with the help of initiation factor, some polypeptide chain will be produced. Now this is the bigger. This is the bigger subunit. 30S, this is say 50S or 60S, as the case may be. So here what happens? There are two sites in the ribosome. Two sides are written, three sides. Let it be two sides. That will be easier. This site is known as A site. This site is known as P site. Why? A site actually P site like this. It occupies this also. A site occupies this also. This is amino acid site. This is known as amino acid or A site. known as amino acid sites. And P site is known as peptidase site. But we will say this A site as arrival site. Arrival site. The amino acids arrives here. Clear? And peptidase site will say this one is parking site. This will say parking site. Parking site. The amino acid gets parked here. There will not be any confusion. But this is amino acid site, this is peptidase site. For our convenience, we will write this as arrival and parking. Clear? Now what happens? Amino acid, this is ribosome, amino acids are coming here. This is initiation of polypeptide chain we are doing, we are going through. What happens? The first amino acids is known as methionine. 1, 2, 3. These have the codon August. This will have the codon August. So August will have the tRNA whenever it comes here. Now the tRNA are coming, carrying the amino acid. So tRNA carrying the amino acid methionine will come here. The first codon tRNA, I will write tRNA methionine. tRNA methionine will come here and they will lie here in the P side, near the P side. tRNA, and you will say something like that. tRNA will come first to P site, where the AUG initiation codon is here. This is the initiation site. So the first tRNA bearing or carrying the methionine usually lies to sit in the P site. Clear? P site, and the methionine is formed. Here methionine will be formed, and there will be a methionine. Methionine will be formed because this is the initiation codon, or initiation codon starts with August. This is the methionine. By this time, the second tRNA bearing the respective or corresponding amino acid along with the amino nucleotide sequence here. Say this is for valine. So tRNA valine. tRNA valine will come here. This is the second one, that is amino acid 2, and this is amino acid 1. So amino acid 2 will come here, and in the methionine, on the amino acid we have seen that in one side it is NH2, C, H, COOH. So this methionine will have COOH, and this amino acid 2, that is what I have said, valine. Valine will have NH2, C, R, H, COOH on the other side. So they will make a CONH2 bond. So this methionine will join with this initiation of polypeptide chain. So polypeptide chain has already started. Here CONH2 bond. So there is a ribosomal movement by this time. Whenever it moves now, there is a ribosomal movement because it moves from here to here. In this way it moves. During this transformation, the amino acid bearing methionine produces methionine, it gets out. It is free now. So it is free or uncharred, I should say. The free or uncharred tRNA gets released. So whenever there is a movement, methionine produced, tRNA, free tRNA is removed, it goes for collecting the further amino acids, and the second amino acid will come to this place, or will be a methionine plus a valine produced, then the third will come, then the fourth will come, fifth will come, and it will be producing like that. So the first tRNA bearing methionine will be at the P site, and the second and subsequent amino acid bearing tRNA will come to arrival site, and they will join with CONH2 bond, and the polypeptide chain is formed. Okay, now we have come to the last part. My question is little different. Say this is 30S, this is P side, this is A side, and the bigger one is it has got P side here. Whenever tRNA, my question is, whenever tRNA bearing methionine comes here, as per the direction of the mRNA, so tRNA comes here, tRNA comes, the first tRNA bearing methionine comes here. Okay, so it will have August, and the anticodon will be UAC, and the tRNA will have methionine. This methionine is produced, clear? This methionine is here. Then it joins with peptide bond with valine, valine with leucine. Now this methionine, listen carefully, this methionine has NH2, C, H, H, COOH, and this valine have NH2, C, H, H, COOH. Clear? Now this is forming a peptide bond. And now after forming this peptide bond, tRNA goes out. Now NH2 site, amino acid sites remain free. Is it not? So in order to produce, say, valine, then leucine, lysine, and say glutamic acid. So 1, 2, 3, 4, 5. So I am producing a particular hormone. That hormone is known as PPSI. This hormone is known as Pepsi. Or pepsin, PPSI, and glutamic acid, then aspartic acid. Aspartic acid. So DNA has given direction to produce pepsin, P-E-P-S-I-N. Pepsin, because these amino acids are in definite sequence. So pepsin will be produced and pepsin will do the purpose of its own digestion. Now this site is remain free. Now if anything happens, if any another amino acid, they are available in the cytoplasm. These amino acids, if any further amino acid gets mixed here, there is amino acid. So instead of producing Pepsi, it may produce Pepsi. It may so happen. So in that case, this area, this NH2 of the methionine of the first tRNA bearing methionine, which occupies AUG, which is the first codon and which is the initiating codon, this is known as, this tRNA is known as tRNA, again very careful, tRNA F met, F methionine or MET met. This first one is known as tRNA F met. It is formilated. So this formylation keeps this side blocked. Clear? First tRNA bearing methionine in respect of AUG or initiation codon is called tRNA F met, as it is formilated and its amino site remains blocked to prevent any further amino acids to get attached and the purpose will be disturbed. So this is formilated. Now the question may come, suppose from aspartic acid, another amino acid known as tryptophan, say for example, some tryptophan, some another methionine. Methionine is there, as per the direction of the DNA, it has produced like this. Like this, it has produced, it has to produce one methionine here. So it will have AUG. And this AUG, if it is filled up by tRNA F met, so there will not be a bond, because the tRNA F met has been blocked at this side. Clear? Clear? Now, to get the proper picture, this methionine bearing tRNA, I am writing methionine bearing tRNA is different with that of the initiator tRNA. This is tRNA F met. This will give it a name known as tRNA M met. So in the polypeptide chain, if there is another methionine or several methionine has to be incorporated because of this protein synthesis, so the other tRNA should be tRNA F met, not tRNA F met. It is formilated. It is known as FORMYLATED. will not go into the deep. This formylation helps the first amino acid. It blocks the first amino acid to prevent contamination with the another amino acid. This is assembly of polypeptide. Polypeptide chain in this way it produces. And now last part is assembly of polypeptide. Clear? Now assembly of polypeptide. After the formation of this polypeptide chain or A, then this tRNA get diffused. The last part is known as after the formation of polypeptide chain. Here we have polypeptide chain prepared. After the formation of polypeptide chain, the RNA, the tRNA get diffused, and after formation this tRNA, the free tRNA, write simply the free tRNA goes out of the ribosome and enters into cytoplasm for collection of further amino acids. This is the entire discussion about this transcription. I have already completed everything. If anything left, go through books, you will get it. And some rho factor and EA factor I have already completed. This sigma factor, rho factor for prokaryotic, and for eukaryotic, no factor is required. Only the last one should have that 3 codons. Because whenever it is being produced from the tRNA, then also there is a direction, it stops, you stop here, you stop here, you stop here, stop codons are there. Whenever it goes until and unless the stop codon comes, and there is the mRNA, whenever mRNA copying mechanism is end, then the new polypeptide chain is formed. This much for the transcription. Thank you very much. Take rest.